Thanks for coming to my talk. I know it's not the best slot, but thanks for spending some time with me this morning. My name is Sebastian. I'm responsible for client and contract management at Wacker Biotech. We are a clinical and commercial phase CDMO, and we also support clients in the cell and gene therapy space. We are active in biopharmaceuticals, but also advanced therapies. So I can't spare you at least one corporate slide. We are part of the Wacker Group. We are 100 years old. We come from the chemicals side. We're headquartered in Munich, and I'm part of the biotech division. Wacker Biotech is a CDMO arm, and we offer process development, contract manufacturing services for GMP manufacturing. So we very much focus on early clinical, late clinical, and commercial phase. We offer recombinant proteins, as I said. Those are mainly used in cell therapy, for instance, as ancillary materials for growth factors. But we also have a dedicated site in San Diego, US, working a lot on plasmids. Plasmids, of course, are crucial for viral vector manufacturing, but they can also be used for non-viral gene therapy systems, also in combination with mRNA. And we have a lot of very exciting projects in this space. We work with big pharma, but also biotechs in Europe, in US, and even Asia in the cell and gene therapy arena. So this gives you an overview of our manufacturing network. We have in total 5 GMP sites, all being EMA FDA approved. In San Diego, we are doing an FDA readiness program at the moment. So we are active in this space since 20 years plus. And all the sites have dedicated process development teams. So we see ourselves as a CDMO with a capital D. So we really want to help you to develop better processes to reduce cost of goods. And I'll have some examples later on. I think what's unique about us, we are typical mid-size CDMO. So we have a strong client focus, a lot of flexibility. But as we are part of a bigger group, we also have access to a central R&D facility in Munich. More than 100 people are working there to improve processes from plasmids, from mRNA, including recombinant proteins. And all of this we apply for the benefits of our clients. Yeah, if you want to have a tour or watch our facilities, you can scan the QR code in the presentation. There's a nice little virtual tour on some of our facilities and gives you a glimpse of what we're doing in our day-to-day. So what's important to me to highlight here, we're not a newcomer in this field. We have released even thousands of GMP batches, some even for commercial, for different modalities. So we come from recombinant proteins. This is our heritage, so to speak. But we are also active in LBP, live biotherapeutic products, a new and evolving, very exciting category, which I'm not going to touch on today. But we also manufacture conventional vaccines. We do a lot of plasmid DNA work. And we've been an early mover in the mRNA LNP space because we manufactured a COVID vaccine mRNA-based for CureVac during the pandemic. And I mentioned we see ourselves as a CDMO with a strong D, and why is that? Because we have a portfolio of platform technologies. All of them are really helping our clients to reduce cost of goods, to improve robustness of processes, and we've just recently extended this into the cell and gene therapy space by offering a platform for plasmid DNA manufacturing, for viral vectors, for instance, but also a platform process for mRNA, which helps you to prevent any kind of IP conflicts. It's a very crowded field, and we want to consult our clients to have, to the extent possible, non-infringing platform process. These are some examples. In the cell and gene therapy space, we work with Helixsmith, for instance. Helixsmith is a Korean-based company working on a pDNA-based gene therapy product. It's in phase 3 currently. And this lead candidate is manufactured in our San Diego site at 650-liter scale. We have 2 undisclosed big pharma projects with mRNA in our Amsterdam facility. I mentioned some have been related to COVID, but also non-COVID applications of mRNA. And we just opened a new mRNA LNP facility in Halle, which is in the center of Germany, so to speak, close to Leipzig and Berlin. Which just received the GMP license. It's part of the German pandemic response, but also half of the capacity is available to CDMO clients. So what do we support? You can come to us with a sequence on paper. We can do the optimization of the genes, of the template DNA, for instance. We can provide you with plasmid DNA for viral vectors, but also plasmid DNA for non-viral gene therapy delivery systems. We worked a lot on mRNA, how to optimize the in vitro transcription, for instance, how to optimize the formulation of LMPs, and we also do fill and finish at our Amsterdam facility for clinical and commercial. So if you look at the typical plasmid DNA manufacturing process, we have branded this technology as Plasmitech. It consists of a set of unique proprietary strains with a focus on complex plasmids, larger plasmids, which you also need in the viral vector space. But of course, we're also, as a CDMO, open to work with your strain and transfer your strain in if you already have one available. We've worked extensively on the upstream to increase productivity and have a reliable upstream process, which then leads into downstream purification. It's a typical 2-column downstream process, followed by the final bioprocessing and filling of the product. And of course, all of this is under the umbrella of our GMP systems. We have quality oversight. We have a process development team at each site helping you to adjust your process to your needs. And I think if someone tells you that every product is the same and you can use only one platform process, you always have to adapt your process. And this is where I think our experts, our scientists, our SME really do a tremendous job in adjusting and tailoring the process to your needs. On the mRNA space, we're also helping our clients with the design of the RNA. It even starts with the sequence optimization. The UTRs, for instance, have a tremendous role in mRNA. We've worked on the template pDNA. For R&D purposes, we can also work with PCR-derived product. For larger scale and GMP, we rely on microbial expression, so fermentation-derived plasmid DNA template. And we also will take this through the in vitro transcription. We have in total 4 lines available in Halle, from clinical supply to commercial supply. The smaller line is typically used for early phase supply, but we can also go up to 30-liter IVT scale. It doesn't sound large if you compare this to microbial or mammalian, but 30-liter in a cell-free reaction is enough even to supply 100 million doses of a product to the market. And of course, we also do purification. It's an exciting topic if you think about how do you purify an mRNA. Most of the processes currently rely on oligo-DT resins, new affinity resins. But if you think about new formats, new mRNAs, circular RNAs, how do you purify them? So also these new modalities and new formats require new ways of purification. And we also do LMP formulation based on either the microfluidic devices or T-mixing. So what we try to do is really to offer our clients with a full package, so also to consult you with potential IP infringements. It's a crowded space. The landscape is getting more and more complex, and as a CDMO, we want to help you with navigating that landscape. To the extent possible, we have a patent-free or non-infringing platform process. I have some slides later on to show you how our process compares to the best in class. And how do we do this? I mentioned a central R&D facility, which is located in Munich. It's outside of our fee-for-service. It's a luxury we have, so to speak, to develop new processes and better processes for our clients. And there are 6 labs working alone just on RNA, including like in vitro assays, functional assays, and also cell-based assays to test the functionality of mRNA formulated in LMPs, for instance. So here's an example. Just wanted to show you at least some data. Our Plasmitech strain is performing very well with regard to upstream productivity. The standard strain here is a typical E. coli strain, public, which is used. A competitor is a best-in-class proprietary expression system, and you see that our Plasmitech is performing as good as the best-in-class. We also work with viral vector plasmids, so you see very nice productivities around 1 gram per liter for some example viral vector plasmids shown in the middle of the slide. And also, if you look at the purity, if you look at the gels, they even do better than standard E. coli strains, and we have a very high productivity if you look at the CC rate of 95% plus. So if you take then your template to the in vitro transcription, I mentioned the crowded IP landscape. Most is related also to capping and tailing of the RNA. There are well-known companies offering their co-transcriptional capping and come with license expectations, so you have to pay then mostly royalties on net sales of your product. Understandably, a lot of biotechs are allergic to this, so we can offer you also a non-infringing alternative, which is the post-transcriptional capping. And you might think that this impacts quality or process time, and I can assure you this is not the case. So it's not more expensive in batch manufacturing, and if you look at the capping efficiency, you can see that we can reach similar titers. Also for longer poly A tails, it works pretty nicely. We have manufactured up to 120A for an mRNA construct, and also the purity is even better than the raw IVT of a co-transcriptional capping. So all of this is available, and we're happy to discuss further with you if this is of interest. Also on the LNP side of things, we are offering for LNP formulation. We're investing into own formulations. We're not yet offering own lipids, but we of course do a lot of R&D work, and we want to help you to be, this is of course part of the process development of your product. LNPs, I think it's crucial if you look at the capping efficiency because of the encapsulation efficiency at the very right of the slide, so you don't want to lose product. So almost or close to 100% capping encapsulation efficiency is pretty good. And the PDI is also an important parameter because it shows you a very narrow particle distribution, which in pharmaceutical development is key for lipid nanoparticle. So I hope that gives you a glimpse that we are also very much driven by science, by technology, and also helping you to improve your product. Last but not least, you also need to prove functionality of your product. And here, if you look at some functional assays of LNP-formulated mRNA, you see that the cells are viable, so they are happy. They luckily take the LNP up. And also you see that in cell-based assays, we see transcription of the gene of interest. And you also see on the right that there's no immunogenicity or adverse immune response. So, and how do we apply all of this? All of this is part of the process development path, and later on it's transferred into our manufacturing network. We have, for pDNA, mRNA, and LNP, several scales available, fitting to all of your needs. We're active in Europe and US, and we're happy to discuss your needs, which facility fits best for your product. And I wanted to mention that also pDNA and mRNA is super exciting in the cell and gene therapy space because there are a lot of very innovative approaches how to deliver a gene. So the non-viral gene delivery, I think a lot of you are aware, is a super hot topic in the industry, and we are working with some players in this field on these new ways of delivering genes to patients. Yeah, to summarize, I hope I could show you that Wacker is not just another CDMO, but we're really driven by technology. We see ourselves as a true partner to develop your product. We have a very long track record. Our SMEs are, of course, crucial for our success and for your success, helping you to develop the best possible process. We are part of a 100-year-old company, still owned by the Wacker family, so we're here to stay. So we're not just another VC-owned CDMO, which is then merged or sold. So we have a very long history with a lot of our clients in clinical and commercial. We continuously invest into our manufacturing network, into our team, into our technologies. And as I mentioned, more than 20 years we're now active in this CDMO space for recombinant proteins, for plasmids, messenger RNA, vaccines. And we also try to supplement this by a set of proprietary technologies. And even outside the pure fee-for-service and vendor business, we are also open to collaborate. We have central R&D, which is, as I said, outside the fee-for-service, where we do also research projects together with university, but also with industry partners. So if you have certain challenges, we also apply here AI tools to optimize processes, to optimize LNP formulations. So we're very much looking forward also discussing with you opportunities to collaborate outside the pure like vendor-sponsor relationship. So, and this is a little cartoon. Yeah, we see ourselves as building bridges. So we try to help you to navigate through all the clinical phases. Not all the bridges are the same. Yeah, some people need a boat, some people need a bridge, some people need a ship for their product. But ultimately, we want to bring better drugs to the patients. And I think this also motivates our team a lot. And you see the guy with the crown, that's always the client. So we also want to do this together with you. And that's why it's not just about science, it's not just about SMEs, it's also about good project management. And this is also part of the responsibility of my team. And if you want to know more, scan the QR code, talk to us. My colleague is here in the forum. We have a table outside. Thank you very much for your attention.